What if a standard black and white CT scan could tell you exactly what your immune system is doing on a microscopic level?
I mean, it sounds completely like science fiction, honestly.
Right. Imagine just looking at that familiar 3D picture of gray shapes, and instead of just, seeing a mass, you could mathematically predict the actual biological warfare happening inside it.
Yeah, but the crazy thing is, it's not fiction anymore. It is rapidly becoming our clinical reality. We're really moving past the era where imaging just shows us anatomy.
Yeah, we're entering an era where imaging reveals the underlying biology, which is exactly what we are getting into today. We are doing a deep dive into a stack of groundbreaking oncology research papers and clinical studies that you brought in.
Right, and it's all focused on this massive leap in precision medicine for colorectal cancer.
Which is huge. Because if you're listening and you follow this space, you probably know colorectal cancer is the third most common malignancy worldwide. And for the longest time, doctors have relied heavily on the TNM staging system, right? So that's tumor size, node involvement, and metastasis.
Exactly. But the problem is, TNM is a fundamentally anatomical classification. It basically tells you the size of the building and whether the fire has spread to the adjacent buildings.
Okay, I like that analogy.
But here is the massive clinical challenge we face. Tumors are incredibly heterogeneous.
Right, they're not all the same. So you could have two patients with, say, stage 3 colorectal tumors. And on paper, under the TNM system, they look identical. Same size, same location.
Completely identical on paper, but their actual biological behavior could be completely different. One could be sluggish, and the other could be wildly aggressive.
Wow. And
Right.
Treating those two completely different biological entities based on just the same anatomical stage.
It's a recipe for poor outcomes. It really is. Because if you undertreat the aggressive tumor, the patient relapses.
Right, which is terrifying.
And if you overtreat the sluggish tumor with systemic, highly toxic chemotherapy, you're causing severe harm for, basically no clinical benefit.
So the goal of this research was to find a way to look at a standard preoperative scan and see the invisible, highly specific risk profile of that exact tumor.
Exactly. And the researchers started with an enormous amount of data. They gathered venous phase CT images from 1,183 patients across 4 different medical centers.
Over 1,000 patients. But they didn't just hand these images to radiologists to eyeball, right? They applied radiomics and deep learning. Let's unpack the mechanics of that, because I feel like it's a huge shift in diagnostics.
It's a massive shift.
Because to the human eye, a CT scan is literally just a gradient of gray pixels. How on earth are they extracting biological meaning from that?
Well, it really comes down to computational power, seeing what human biology simply can't. So radiomics is the high-throughput extraction of quantitative features from medical images.
Okay, so breaking that down a bit.
When a human radiologist looks at a tumor, they see shape, they see margins, maybe some general density. But radiomics looks at the exact mathematical relationships between the pixels.
Wait, so it's doing math on the grayscale.
Yeah, exactly. It calculates spatial heterogeneity, subtle texture patterns, the microscopic contrast between adjacent areas of tissue. It basically turns a visual image into a massive, highly dimensional numerical data set.
So it's finding a hidden mathematical fingerprint in the static.
That's a perfect way to put it. And then the deep learning component takes it even further. The papers detail the use of a 3D convolutional neural network, or CNN.
And they pre-trained that on a massive medical data set called Med3D, right?
Yeah.
I think it's worth highlighting the 3D part here. We're not just scanning flat pictures like a photograph.
No, and that's a crucial distinction. A traditional 2D network just looks at pixels on a flat plane. A 3D CNN looks at voxels, which are volumetric pixels.
Voxels. So it understands spatial depth.
Yes. It allows the algorithm to comprehend the tumor as a dense spherical architecture. It analyzes how the tissue changes from the core of the mass all the way out to the invasive margins.
Wow. So it doesn't just measure predefined textures like radiomics does.
Right. It actually learns its own features by finding hidden, abstract patterns through multiple layers of artificial neurons.
Okay, so between the radiomics and the deep learning, the sources say the researchers initially pulled 1,437 radiomics features and 512 deep learning features from these tumor images.
Which is an overwhelming amount of data.
I mean, that is nearly 2,000 distinct data points for a single tumor. But from a data science perspective, you can't build a practical, generalizable medical tool with 2,000 variables, can you?
No, absolutely not. The model would overfit immediately.
Meaning you would just memorize the training data and then fail in the real world.
Exactly. You're dealing with extreme dimensionality. The signal is just buried under a mountain of noise. So to solve this, they systematically evaluated 117 different machine learning algorithm combinations.
117, just to find the absolute best method for filtering the data and predicting overall survival.
Yeah. And the winning combination was a pairing of lasso and GBM, which ultimately created their core model. They called it the DLRM-.
Which stands for Deep Learning Radiomics Model-Based Risk Stratification. Quite a mouthful.
It is, yeah.
But let's break down how that lasso and GBM pairing actually works. So I know lasso stands for least absolute shrinkage and selection operator. I always picture like a cowboy with a lasso standing in front of a stampede of 2,000 data points.
I love that image.
Right. He's swinging his rope and only catching the absolute strongest, most predictive features while the rest of the herd just runs past.
It's actually a really effective way to visualize it. Mathematically, lasso applies a penalty to the model's coefficients.
So if a feature doesn't strongly correlate with patient survival,
lasso shrinks its value exactly to 0. It effectively eliminates it. It is a ruthless editor.
wow.
Yeah, it pared those nearly 2,000 features down to just 15 highly predictive variables.
Only 15 out of 2,000. And then they fed those 15 surviving features into GBM, which is gradient boosting machine.
Sure.
If lasso's doing the filtering, I assume GBM is doing the heavy lifting for the actual prediction.
That's exactly right.
Is it basically a sequence of decision trees where each tree learns from the errors of the previous one?
That's the underlying mechanism, yes. It's an ensemble learning method. So it builds a weak prediction model, evaluates where it made errors, and then builds a second model specifically designed to correct the residual errors of the first one.
So it just keeps iterating.
Right. It incrementally boosts the accuracy until it converges on an incredibly sharp, refined prediction.
Okay, so let's just recap where we are for a second, because this is dense. The lasso filters out the noise, and the GBM iteratively corrects its own homework.
Yep.
And the result is a model that uses just 9 radiomics features and 6 deep learning features. Just 15 mathematical points from a CT scan could accurately stratify patients into high-risk and low-risk survival groups.
And they proved it rigorously across the training cohort and entirely separate external validation cohorts.
But the researchers didn't just hand this AI score to doctors and call it a day, did they?
No, they knew better than to operate in a vacuum. A sophisticated AI model is powerful, sure, but clinical reality involves integrating new technology with established medical parameters.
Which brings us to the nomogram. So they performed multivariable analysis and found that integrating their AI risk score with traditional markers created an even more powerful predictive tool.
Specifically, they integrated it with CEA levels and end stage.
Right. And just for context, CEA is carcinoembryonic antigen, which is a blood protein, and end stage is the lymph node count. But I have to admit, when I was reading through these sources, this felt a bit like putting training wheels on a Ferrari.
How do you mean?
Well, if the deep learning model is analyzing thousands of volumetric data points and finding these invisible mathematical truths, why do we need a basic blood test and a lymph node count to prop it up? Isn't the AI smart enough?
It's not really about propping it up, though. It's about capturing entirely different dimensions of the disease process.
Different dimensions.
Yeah, so the AI is incredibly adept at analyzing the primary tumor's internal chaos. Its local heterogeneity, the immediate density, the localized biological behavior.
Okay, so the local area.
Right. But end stage and CEA provide systemic context.
Ah, systemic. So it's like a highly advanced weather forecast. The AI radiomics model is measuring the atmospheric pressure and the temperature right over the city. It's telling you the exact potential for a massive storm to form right there.
Yes.
But the end stage and the CEA, that's the radar showing you that the storm has actually already started moving and the wind is already blowing at 50 miles per hour outside the city.
That captures the dynamic perfectly. You really need both to predict the ultimate impact. The AI measures the aggressive potential of the primary site, while the clinical markers measure how far the disease has actually progressed systemically.
So they combine them into a clinical nomogram. And a nomogram is essentially a graphical calculating device that gives doctors a customized storing chart.
Right. Exactly. And by doing that, they created a tool that significantly outperformed both the standard clinical model and the standalone AI model in predicting 3-year and 5-year overall survival.
It essentially gives the doctor a highly personalized, incredibly accurate biological compass.
It does.
But here's where the research gets truly fascinating for me. Because an AI doesn't know what cancer actually is. It doesn't know what a cell is. It only knows mathematics and voxel contrast.
Right. It's just numbers to the computer.
So what biological reality is actually creating that math on the scan?
Right.
Why does the AI flag certain gray blobs as high risk and others as low risk?
This is where the study completely bridges the gap between computer science and molecular biology. To understand the why, they performed transcriptomics on tissue samples from a subset of the patients.
Transcriptomics. Okay, let's break that down. That's looking at RNA, right? If DNA is the master blueprint of the cell, RNA is the active work order being sent to the factory floor.
Exactly. By looking at the transcriptome, the researchers could see exactly which biological processes were turned on or turned off in the tissue at that exact moment.
Which is incredible. And what did they find?
The gene set enrichment analysis revealed two completely different biological universes when comparing the high-risk tumors to the low-risk tumors.
So the AI risk score perfectly mirrored different biology.
Yes. In the high-risk tumors, the ones the AI flagged for poor survival, the RNA was heavily enriched in extracellular matrix, or ECM pathways.
The sources noted specific pathways like collagen fibril organization. And the extracellular matrix, for those listening, is essentially the scaffolding between cells.
Right, it's the structural support. And in these high-risk patients, the tumor is actively building a massive, stiff, protective structural fortress around itself.
Wow. It is literally laying down thick collagen cables.
Which creates a very hostile environment for the patient. A stiff, dense ECM does several things. It promotes aggressive tumor cell migration.
Okay, so it helps the cancer spread.
Exactly. It also alters tissue mechanics in a way that drives malignancy. And crucially, it creates a physical, dense barrier that prevents chemotherapy drugs and the body's own immune cells from penetrating the tumor core.
So it's building a microscopic medieval castle wall.
That's exactly what it's doing.
Now contrast that with the low-risk tumors. The ones the AI predicted would have high survival rates. Their RNA profiles were heavily enriched in immune-related signatures, right? We're talking pathways for antigen presentation, immunological synapses, and massive infiltration of CD8 plus T cells.
Right. And CD8 plus T cells are the cytotoxic assassins of the immune system.
The assassins, I love that.
Their primary job is to hunt down and destroy compromised cells, including cancer cells. So in the low-risk tumors, the patient's immune system has successfully invaded the tumor microenvironment and is actively waging war.
Which is great news for survival.
Yeah.
But wait, this brings us right back to the central mystery here. The CT scan is macroscopic. It's looking at millimeters of tissue at a time. The immune cells and the collagen fibers are microscopic. How on earth does a macroscopic CT scan see a microscopic immune response?
Well, it sees the physical consequences of that biology. Microscopic cellular processes eventually create macroscopic structural changes.
Oh, I see.
Yeah. The dense pack-in of a collagen fortress, the swelling and inflammation associated with a massive immune cell infiltration, the chaotic, leaky blood vessels that feed aggressive tumors, all of these fundamentally alter the radiodensity, the water content, and the texture of the tissue.
Oh, wow. So the AI isn't seeing a T cell. It's reading the volumetric shadow that a million T cells cast on the tissue's physical structure.
Exactly. It's measuring the architectural difference between a dense collagen wall and an inflamed immune battlefield.
That is just brilliant. Okay, so let's do another quick recap. The AI finds a pattern. The RNA proves the pattern is real biology fortresses versus immune battles. But the sources showed the researchers didn't even stop there. RNA tells you what the cell is trying to do, what the factory orders are. But they wanted to know what the cells were actually doing in real time on a chemical level.
Which requires metabolomics.
Metabolomics. So when cells burn energy, build those collagen walls, or fight immune wars, they process chemicals and leave behind tiny byproducts called metabolites.
Right, it's like cellular exhaust. And to measure this chemical exhaust, the researchers used NMR spectroscopy on paired samples of tumor tissue and adjacent normal tissue.
NMR spectroscopy. Nuclear magnetic resonance spectroscopy. It's a fascinating technique.
It really is.
It basically uses powerful magnetic fields to interact with the magnetic properties of atomic nuclei in the sample.
Exactly, because different chemical compounds resonate at very specific, predictable frequencies.
It's like tuning a radio to specific chemicals, right? By reading these frequencies, researchers can perfectly identify and quantify the exact concentration of various chemicals in the tumor.
And the chemical fingerprint they found was remarkably clear. They identified 17 key metabolites that linked the tumor's development directly to the AI's risk stratification.
17 metabolites.
Yeah, and most importantly, 2 specific metabolic pathways emerged as the critical drivers in the low-risk group, meaning these pathways are highly protective.
And those two pathways were butyric metabolism and nitrogen metabolism. And I noticed the researchers were really meticulous here. They didn't just trust their own local patient samples, do they?
No, they knew they needed external proof. So they validated this specific chemical finding against an entirely independent, massive public database.
The Cancer Genome Atlas, or TCGA.
Exactly. They checked 417 independent patients and confirmed it. Patients with lower activity in butanoate and nitrogen metabolism had significantly worse overall survival.
The TCGA validation is really what elevates this from an interesting localized finding to a robust, generalizable biological truth. But I want to zero in on butanoate for a second, because that term really jumped out at me.
Oh, yeah.
Yeah, because in the broader nutrition and longevity sciences, butanoate, which is usually referred to as butyrate, is a short-chain fatty acid predominantly produced by our gut microbiome when we digest dietary fiber.
That's right.
Are these sources suggesting that a patient's gut bacteria might be fundamentally altering the chemical microenvironment of the tumor, making it less aggressive?
The data strongly points in that direction, yes, and the mechanism is well documented in broader oncological research. Butanoate isn't just passive energy. It functions as a histone deacetylase inhibitor, or HDAC inhibitor.
Okay, let's explain what an HDAC inhibitor does for the audience.
So it effectively alters gene expression. In the context of the tumor microenvironment, butanoate acts as a powerful anti-inflammatory. And critically, it prevents CD8 plus T-cell exhaustion.
Wait, really?
Yeah.
So when the cytotoxic assassins, the T cells, are fighting the tumor and starting to burn out, the butanoate swoops in, alters their gene expression, and forces them to keep fighting.
Precisely. It sustains the immune response, and the nitrogen metabolism works in tandem with it. Well, while nitrogen is often associated with cellular growth in a balanced tumor microenvironment, proper nitrogen metabolism is essential for maintaining redox balance.
Redox balance, meaning the balance of reduction and oxidation. It's basically managing the toxic oxidative stress, the free radicals.
Exactly. The tumor microenvironment is incredibly toxic and stressful. If the redox balance fails, the immune cells fry themselves.
Ouch.
Nitrogen metabolism helps manage that stress, keeping the environment hospitable enough for the CD8 plus T cells to actually do their job.
So when the AI's 15-feature algorithm looks at a scan and categorizes a tumor as low risk, it isn't just saying the cancer is lazy.
Not at all.
It is identifying a highly specific, complex metabolic state. It's looking at a tumor where the patient's own body, fueled by healthy metabolites like butanoate, is actively and successfully suppressing the malignancy.
Yes. The AI is reading the terrain of the battlefield and mathematically proving that the patient's immune system is winning.
It is just a profound synthesis of data. I mean, we've gone from the macroscopic pixel shading of a CT scan down to the microscopic collagen fibrils of the extracellular matrix, all the way down to the atomic resonance of butanoate molecules.
And they all tell the exact same story about patient survival.
Which brings us to the ultimate question. What does this mean for the person actually sitting in the hospital waiting room? For anyone listening who works in healthcare or, who has a family member navigating this disease, this research really feels like the blueprint for the next decade of precision medicine.
It really does. It represents the end of the one-size-fits-all approach to oncology. If a routine, non-invasive CT scan can accurately identify the biological reality of the tumor before a scalpel ever touches the patient.
It changes the entire treatment paradigm.
Completely.
We can spare the low-risk patients, the ones whose bodies are already fighting back successfully, from the harsh, toxic systemic damage of unnecessary chemotherapy.
Exactly. And conversely, for the high-risk tumors, the ones rapidly building those dense collagen fortresses, doctors can immediately deploy their most aggressive, targeted therapies without wasting precious time.
It allows us to match the intensity of the treatment to the exact intensity of the disease, mapped specifically to the individual patient's biology.
It's personalized medicine at its finest.
And as we wrap up this deep dive, there is one final, incredibly provocative thought from these sources that I want to leave you, the listener, with.
Oh, the microbiome connection.
Yes. If the patients who survive, the low-risk group, have highly active butanoate metabolism inside their tumors, and we know that butanoate is heavily produced by the gut microbiome fermenting dietary fiber, could the future of cancer treatment begin in the gut?
It really opens up the possibility of metabolic prehabilitation.
Prehabilitation, exactly. Could we see a future where the moment a tumor is detected, the first prescription isn't a chemical, but a highly specific personalized diet or targeted probiotic regimen?
It's a huge area of ongoing research now.
If the food we eat alters our gut bacteria, and those bacteria pump out specific metabolites. And those metabolites dictate whether our immune cells have the energy to break down a tumor's fortress. Could we intentionally alter the tumor's microenvironment before surgery?
It's entirely possible.
Could we literally eat our way to a lower AI risk score? It seems the diagnostic tools of tomorrow won't just tell us if we are sick. They will read the incredibly complex, muddy waters of our biology, tell us exactly how our body is fighting back, and perhaps tell us exactly what to feed the troops.